So now when we do muscle physiology, I purposely had this image up here. We're going to do this much later, but at a point when we are going to do this image, this part, which is known as excitation, contraction, coupling, I'll kind of refer you to this. So it's a good idea to kind of print this out as one slide so that you can refer back to it. It gives it quite well. So let's first begin with how we classify muscle. So again, like everything else we've been doing before, we always classified, we classified bones, we classified tissues. So now we classify muscles, and we classify it in two ways. One is a functional classification, and that's based on whether it's under your control or it's not. So when it's voluntary, then we call it skeletal muscle. And you'll see later that the voluntary, anything which is voluntary is supplied by a part of the nervous system which is known as the somatic nervous system. So this is a good time to kind of get used to some of these words. So it is the nerve supplies through the somatic nervous system. So nerve supply is through the somatic nervous system. So in other words, the somatic nervous system is a voluntary nervous system. It controls any voluntary activity. And functionally, any muscle which is not under your control is called involuntary, and the examples are cardiac and smooth, and that makes sense. You can't control your heart rate, and you can't control the activity of your smooth muscles which are found in the walls of organs. For example, the stomach. You cannot control the contraction of your stomach or your urinary bladder or other things. So these are made of smooth muscle. So this therefore is involuntary. So this is not supplied by the somatic nervous system. Instead, it is supplied by the autonomic nervous system. So autonomic, you can understand, would also be called the involuntary nervous system. And there are two parts to the autonomic. These are called parasympathetic and sympathetic. We will do these later. You've heard one of them, sympathetic. You've heard when we did the skin, for example. Remember, I told you all the erector pylorus muscle was supplied by the sympathetic nervous system. That was smooth muscle. So you can see how, that's not under your control. So this autonomic nervous system supplies both cardiac and smooth because they're involuntary. The other classification we have is structural. So when you're looking at it under a microscope or even electron microscope or a regular microscope, there you find some muscles have a banding pattern. That banding pattern is known as striated or banding pattern, where you can definitely see dark and light. So it's kind of like little lines. Stria means lines. So structurally, skeletal and cardiac both show a banding pattern, whereas smooth muscle is smooth, so it doesn't show any banding pattern. So it's called non-striated. So no lines are seen. So very simple classification. So you sometimes will be asked to put the two together. For example, in this question. So let's answer this question. How would you classify cardiac muscle if you had to use both functional and structural classification? Okay, well, most of you got it right. Yes, it is involuntary and it is striated. So again, if we look back, so it's not involuntary and non-striated. It's not voluntary and non-striated at all. And it's not voluntary and striated. Remember, it's not under your control. So then you automatically think involuntary. So if you look up here at this slide, you go back here and look at this, see this. Involuntary is cardiac. And again, in striated is cardiac. So it's both striated and involuntary. Okay? So that's how you're going to put the two together. So this would hold true for the other muscles too. So you must know, if you were to classify skeletal, how would you classify it? If you were to classify smooth, how would you classify it? So make a little note for yourselves to kind of classify them that way. Now let's look at some properties of muscles, and this is quite easy. Muscles are excitable. They respond to stimuli. So, you know, when a nerve impulse comes and stimulates the muscle, it reacts. Muscles are capable of shortening, and that's contraction. So sometimes you will find that muscles don't necessarily always have to shorten when they contract. So, you know, in layman's term, when you say contract, you use the word shorten. But you'll see sometimes here, when we talk of muscles contracting, that's because of a relationship between filaments inside the muscles, and they kind of, you know, attach to each other. But most of the time what happens is, when these filaments attach to each other, they also cause the muscle to shorten. So contraction and shortening does not necessarily mean always the same thing. You will find, and I'll show you, I'll give you an example where you will have muscles where they are contracting but they are not shortening. The length remains the same. And sometimes you'll have a situation where the muscle is actually contracting but it is lengthening at that time. But here, one of the properties is, this is one of the tissues in the body which can actually shorten. Okay, so that's the contractility power. They can elongate also. You know, muscles can stretch. And in fact, when you go to the gym, you actually want to stretch before you begin your exercises, because muscles tend to shorten by 2/3 of their resting length. So if the resting length is more, so if you stretch a muscle before you begin your exercises, so your resting length is more, so you have more length whereby you can shorten it more. So that's the purpose of stretching. Then muscles can also recover. So when muscles shorten, they can kind of recover and they go back to their original size. So that is elasticity. And this happens in many parts of the body, you know, like your stomach, for example. You all know this obviously of skeletal muscle. Your muscle contracts, you pick up an object and then it goes back to its relaxed state. But even your stomach, let's take an example, or your urinary bladder, that fills, that stretches. And then when your urine is voided or your stomach, your contents have gone out, the stomach comes back to its normal size, right? So it's capable, as you can see, of stretchability and elasticity, of coming back or recoiling to its normal size. Here are some movements of muscles which you already know. They produce body movements. You know, muscles are attached to bone and they produce movements. They stabilize your position. They maintain posture. We have certain very important muscles in our back which maintain your erect posture. They stabilize the position of joints. They produce movements because they are attached to bones. They regulate organ volumes. I just gave you an example of how, you know, in the stomach and the urinary bladder, they can stretch and then they can contract. Of course, they can stretch to a particular limit, after which you begin to feel pain. You know when your bladder really fills to a great amount, you begin to feel pain and you just have to go. And you will find that and this is especially true of smooth muscle, that in areas of the body, smooth muscle becomes highly and it's kind of arranged in a circular fashion. So it becomes quite thick like this, and it forms a valve. That valve is known as a sphincter. So another name for a sphincter is a valve. And you will find these sphincters in areas of the body where things go from one area to the other, or from one part of the body to the exterior. So from one part of the body to an exterior, I'm giving you an example like the urinary bladder. You know, when your bladder fills, you all know urine doesn't pass out because the bladder is closed at the end. So if I was to draw a picture of the urinary bladder like this, at the neck of the bladder, there's actually a little sphincter present here, and then this part is known as the urethra. And here also there is a sphincter. Whenever smooth muscle forms a sphincter, that is involuntary. Remember, smooth muscle was involuntary. Whenever skeletal muscle forms a sphincter, that is voluntary. Okay? So as I said, most of the times these sphincters are formed by smooth muscle, but in some parts of the body you have both smooth and skeletal, and urinary bladder is a good example. So here you have an involuntary sphincter, so you have no control over it, so it just keeps it closed, the bladder keeps filling, and then after some time what happens is when it fills to too much, that's when you feel pain and then you desire to void. So the nervous system will act and it will cause this to open. And then you also have a voluntary sphincter. And you'll see this in the bladder. This is also present in the anal canal. Because if the time is not right for you, your voluntary sphincter will not open. Because you don't just go pee anywhere, right? So the time is not right for you, you won't go. So you have these kind of circular muscle fibers which are tight. They act like valves in that area. When they are smooth, they are involuntary. When they're made of skeletal muscle, they are voluntary. Okay?
Is one stronger than the other?
Is one stronger than the other? I can't say whether one is stronger than the other. No. They're both equally strong. It's very difficult. They can be overridden. So no, I can't really say one is stronger. They can be overridden because if your bladder fills beyond a particular amount, the pressure is so much that it wouldn't matter.
How much you're saying, no, I don't want.
Yes, exactly. So so one is not stronger than the other. Then muscle functions, they also move substances within the body, and all of these is usually to do with smooth muscle. Not skeletal, this is smooth. So, for example, your blood vessels, they have smooth muscle in them. Lymph vessels have smooth muscle. I just showed you the urethra in the case of the urinary bladder. You also have ureters which bring urine to the bladder. Your esophagus is partly skeletal, but otherwise the small intestine, all of the gastrointestinal tract, most of it is smooth. The reproductive tract, that's again smooth muscle. So here, the contraction of the smooth muscle allows substances to move across. So you can see how they move substances within the body. They also produce heat, so, metabolic activity. So you have when you feel cold, you shiver, and that's the skeletal muscle shivering, and that produces heat, and that's what keeps you warm. And then you've also seen, this doesn't produce heat, but you've also seen the erector pylorus muscle, which we did in the integumentary system. You remember it when it contracts, the hairs stand up and it kind of traps heat. Not such an important function in us, but in animals it's more important, right? But in us, it kind of helps to squeeze the sebaceous gland so that, it lubricates the hair follicle. So again, you can see another function of muscle. And then muscles, because they act across joints, when they cross a joint, they act on it, they help to also stabilize the joint. So they make sure that the joint kind of moves in the proper fashion. So that's another very important action of muscle. When the muscle is paralyzed, usually you will have some joint problems also. Now let's look here in this lecture. We are going to describe, most of what we are going to talk about is skeletal muscle. Okay, so from here on, all I'm talking about really is skeletal. If I talk about smooth or cardiac, I'll let you know. So skeletal muscle, when you look at the gross anatomy of skeletal muscle, as I said earlier, its nerve supply is from the somatic nervous system, which is the voluntary nervous system. And in the somatic nervous system, either in the brain or in the spinal cord, you have areas where nerve cells are collected. So that is known as gray matter. So gray matter or nuclei are areas where nerve cells are collected. So we can say collection of nerve cells is known as gray matter or nuclei. And this image here is showing you the spinal cord. So here you can see the spinal cord. So this area, this H-shaped area in the spinal cord is called gray matter. And from here, nerve fibers will start, a neuron will start, which is a nerve cell and its process. And that goes and supplies the skeletal muscle. So you can see. So this is for the skeletal muscle. Smooth and cardiac, as I said, they are supplied by the autonomic nervous system. When we do the autonomic nervous system, we'll kind of go into more detail about that. And muscle is also very richly supplied with blood vessels. So blood vessels usually will enter the center of a muscle. And that center of a muscle is also known as the neurovascular hilum. The word hilum, get used to this word. The word hilum means gateway or entry point. So the word hilum means entry point or gateway. Neurovascular would mean nerve and blood vessels, right? Neuro for nerve, vascular for blood vessels. Now our body is extremely economical. It doesn't do anything, nothing unnecessary is done. So imagine if there's a door through which a blood vessel is going, it decides, why should I open another door and make the nerve go through there? So instead, all blood vessels and nerves go through the same area. So that's why that opening is called the neurovascular hilum. Can you see, like, it kind of conserves tissue? Why, make another hole in the blood, in the skeletal muscle to go from that area? So when you, if you ever get an opportunity to look at a cadaver, you will find whenever you're looking for a nerve, you often see the blood vessels first, and then you can trace them and you'll see a nerve along with it, because most of the time the main blood vessels and nerves enter together at this neurovascular hilum. Okay? So this image is kind of trying to show you that, but it's a little blurry, so you can't see. Now here I also want you to look at a particular term which we will come to later, but I'm going to kind of introduce it here, which is known as the motor unit. So look up here. Okay? And what you can see here is, you can see, as I said, a skeletal muscle receives its nerve supply from the gray matter of the spinal cord. So there's a single nerve fiber. Don't write this down, we'll look at it later, but I just want to show you what this is so that you can refer to it later. So here is a motor neuron. That means a single nerve fiber, a single nerve cell. It comes out, and can you see it is supplying not just one muscle fiber, but or one muscle cell, but it's supplying 2 muscle cells here. This red one is supplying 3 muscle cells here. Can you see that? So that is known as a motor unit. In other words, a single neuron and the number of muscle fibers it supplies is known as a motor unit. So it's like a unit, they work together. So muscle cells on one side and the nerve cell which supplies it together, they have to work. You need both to be functioning in order for the muscle to be able to actually act. Okay? Now let's look at again, part of gross anatomy. The muscle is covered by connective tissue. And here's where we are going to draw on all that we've been doing in the past. So remember, we talked of connective tissue. And if you remember, I talked about loose connective tissue and dense connective tissue and loose or areolar tissue. I told you it's like forms packing material. So the next time you go to the grocery store and you buy a steak, you'll see in the steak, you see, when you look at a steak, I'm sure everyone has seen this, those of you who eat meat. So imagine you had a nice big steak like that. Okay. So you all know, a steak you can kind of make out little areas like this, right? This is about what you can make out in a steak, okay? So this is the whole muscle, okay? This whole thing is the whole muscle that you have. And then inside here, these little bundles that you have, these bundles are known as fascicle. Fascicle means a bundle, okay? That's all you can actually observe in a steak. But if you were able to observe it a little bit better, you would actually notice, suppose you could really see, which you can't, inside each of these bundles of fascicles, you actually have, it is in turn made up of single cells which are known as muscle cells here. Okay? So we have the whole muscle. Then the muscle is broken up into bundles. And each bundle actually inside it has cells. Muscle cells are also known as muscle fibers. Do not confuse this with, so far you've been doing, whenever you talk about fiber, you think of something thin and long. It may run in bundles or it may run singly. And what are the fibers you know of? Collagen fibers, elastic fibers, right? You know of these, and reticular fibers, okay? What were they? They were just sort of strands. They didn't have any nucleus or anything inside, okay? When you think of cell, and if we drew it, and do you mind if okay, let me draw it here. So here, if you think of a cell, either you'll think of a cell which looks like this, or you think of a cell which looks like this if it's cuboidal or columnar, or if it's squamous, right? You think of a tiny little cell like that. When you look at a muscle is not a tiny cell like this. It actually is a very, very big, long cell. And I'm talking about skeletal muscle here. It's a very long cell. So the cell, because it's long, we also call it a fiber. Okay? So this is a muscle cell, or also known as a muscle fiber. So remember that.
So where would the nucleus?
So in the case of skeletal muscle, it actually is made up of many cells. So these were many cells which fused to one another, and these cell outlines broke. So now you got this long cell. So it is multinucleate. If you remember from histology, a skeletal muscle is multinucleate, many nuclei, and these nuclei are present just below the cytoplasm, which we say was hypolemmal nuclei. So it's called many nuclei like this. Okay? So now if we use this same thing and we say this whole muscle, which is here, this your stake, that is covered by a connective tissue covering, and that connective tissue covering on the outside, so here this is this whole muscle covered by connective tissue, that covering is known as epimysium. So how are you going to remember it? If you remember the word epi and think of epidermis, remember epi was on the outside, right? Epidermis was the outermost layer of your skin. So similarly, epimysium, mysium is for muscle. The word mysium, epi is on the outside, okay? Then this epimysium actually sends in septa to surround each fascicle. So you can see these were the fascicles. So here he's pulled out one fascicle and shown you. So covering the fascicle, there's a connective tissue layer which covers the fascicle. That covering is called perimysium. So you can see peri meaning going around. Okay?
So the one piece that's coming out.
It is just showing you one fascicle. So all of these are fascicles. So covering each fascicle. So all of this would be perimysium. So this epimysium is the one which actually sends perimysium around this, sends perimysium around this, around this, around this. So each fascicle has its own perimysial covering, okay? The epimysium is just one because it covers the entire muscle. Then inside each fascicle, if you remember, I said you had muscle cells. So here are muscles. This is a muscle fiber which has been drawn out, or a muscle cell, this whole thing. And here you can see the nuclei. So this whole thing, which is this, which I'm showing you, these round things, each muscle cell or muscle fiber in turn is covered by a layer which is known as endomysium. So they haven't labeled it. So I'll label it for you. So here muscle fiber covered by endomysium. Oh, here it is, sorry. Endomysium between individual muscle fibers, or wrapping around each individual muscle fiber. So this part here would be endomysium. So this would be endomysium. This would be perimysium, and this would be epimysium. And if you remember, endo, the word endo means inside, endosteum, remember, lining the medullary cavity, so endothelium, okay, so endo is covering the muscle cell, epi is covering the whole muscle, and perimysium is covering the bundles, okay? And the way you can remember is epi on the outside, endo on the inside, and peri in between. Okay, so the two E's on the outside and the P in between. Okay. Now here's where it gets a little bit more confusing. So this is what is kind of a little bit seen in if you look with the naked eye. With the naked eye, you can see the whole muscle. Like I said, in the stake, you can see these bundles. You may not be able to really see these individual muscle cells. But let's say that, assume that you can. Then if you went a little bit more microscopically inside, you will find that actually each individual muscle cell, so this is one muscle cell. Can you see it is kind of made up of thin fibers inside, smaller fibers. So the word fibril means even smaller than a fiber. So inside each muscle cell, you have many myofibrils. So here, if I had to draw myofibrils, I'll say that these were myofibrils. Okay, so each muscle cell is made up of many myofibrils. Myofibrils do not have their own covering. Okay, so it's only muscle, fascicle or bundle, and a muscle fiber or cell which has the covering. Beyond that, there are no separate coverings. So the myofibrils don't have any named covering of their own. So now what we are going to do is we are going to go from the muscle to the myofibril, and to make things even more confusing, the myofibrils are in turn made of something else, okay? So, but I want you all to kind of think first big like this, and then that's why looking at these pictures becomes really important. So which is why I had this nice slide from your book up here. So here's how we divide it. So first we have the big muscle, which was covered by epimysium. And here you have it, see, covered by epimysium. The muscle then divides into a fascicle. So there are many fascicles. Each fascicle is surrounded by perimysium. Each fascicle is in turn made up of many muscle cells or fibers, and each cell is surrounded by endomysium. So here it's showing you a single cell, a single muscle cell. So here there are many muscle cells. So here it's showing you a single muscle cell. This muscle cell is made up of myofibril. Remember the word fiber, and fibril means even smaller. It's like duck and duckling. I can't quickly think of any other example, but fiber and fibril, okay? So myofibril, so each muscle cell is made up of many myofibrils. So here you can see that. And inside the myofibril, we said muscles were striated. Remember, some cardiac and skeletal muscle was striated or banded. So what actually gives rise to that banding pattern? That is because of some proteins which are present inside, which are known as myofilaments. Okay? So myofilaments are the actual unit which is responsible for the shortening and contraction of muscles. So when we see that, we'll be talking about these myofilaments, and they have different names. So again, to review, whole muscle made up of many bundles called fascicles. The fascicle in turn is made up of many muscle fibers. So this is showing you one fascicle and many muscle fibers. Here it's taken one muscle fiber and then shows you that each muscle fibers actually has little threads inside it, which are known as myofibrils. And these myofibrils are actually, they have protein filaments inside them, which we'll see later, and these are known as myofilaments. So you have something called a thick filament and you have something called a thin filament, okay? It's only up to here that we have coverings. Notice, only up to the muscle fiber you have coverings. These parts, the myofibril and myofilaments, don't have any coverings of their own, okay? Now, let's answer this question as a review. So this is one kind of question, which means that you must know what epimysium covers, you must know what perimysium covers. Good, muscle fiber. Remember endo, think of endosteum, inside. The muscle is the big picture. So it will be covered by epimysium. Fascicle is the next big thing. It's got the P. Fascicle is covered by the perimysium. And fiber is the one which is covered by endomysium. Okay, remember, myofibrils don't have any coverings. So now we are going to look at this myofibril. So here's where the physiology, what is it that actually causes the contraction of muscle? We've looked at all the big picture, right? And we'll look at it more when we do the muscular system, how the whole muscle contracts and all. Now we're going to see what is the thing that actually causes the muscle to contract. So here, I'm looking at this is one muscle fiber which has many myofibrils. Can you see that? So this is one muscle fiber showing many myofibrils. So that's why looking at these pictures and reading everything is really, really important. So let's look at some terms that we use. Anything to do with muscle, we use the word sarco or we use the word myo. So you saw like myofibril, myofilament, like that. Or we often also use the word sarco. So the cell membrane, because the muscle is like any cell, right? Its muscle fiber is a cell. A cell has its cell membrane, or what we call plasma membrane. Now in the case of muscle, this cell membrane, instead of calling it just plain plasma membrane or cell membrane, it's given a special name, and that name is sarcolemma. So see, use the word lemma, and you see the use of the word sarco. So it's called sarcolemma. The cytoplasm in the muscle is called sarcoplasm. And in that cytoplasm are these myofibrils, you have mitochondria, you have the nucleus, you will have Golgi body, all of that will be present. So sarcoplasm is the cytoplasm. You have many nuclei. Remember I said skeletal muscle was one long cylindrical fiber, and you also did it when you were doing tissues. So you have many nuclei. These nuclei are present just below the sarcolemma or cell membrane. So that's why we say the position of the nucleus is hypolemmal. The word hypo means below. Whenever we use the word hypo, it means below. So below lemma or below sarcolemma. So the nuclei position is below the cell membrane. So which you can see here. So here's one nucleus, here's another. So that's why when I drew that muscle fiber like this, if I draw a muscle fiber, this is where the nuclei are going to be, like, you know, ants in a line, just below the sarcolemma. Like any other cell, it has mitochondria because muscle needs energy and it uses oxygen to, you know, produce ATP. So lots of mitochondria are present. So here are some mitochondria seen. It doesn't show all the other cell organelles, which we'll see. It will have endoplasmic reticulum, which the name will be Sarcoplasmic reticulum instead of calling it endoplasmic reticulum. So it has all of that too. Then, as I said, each muscle cell is made up of many myofibrils, right? So here you can see one muscle cell, many myofibrils, and if you pull one myofibril out, this actually is made up of little proteins which give it this banded appearance. Those proteins are known as myofilaments. So the myofibril has myofilaments which give it that banded appearance. You can see a dark and a light, dark, light band. And those bands we will see are known as A and I bands. Those bands are called A bands and I bands. The A band are also called dark bands. A are also known as dark bands, and they come from the word anisotropic. The A gives it A bands. The I bands are the light bands, and they are from the word isotropic. So that's why we use the first letter and I band. Okay? So A and I bands are made up of myofilaments. And this myofilament is what is seen in the myofibril. Okay? So now we are going to, so we did the myofibril, so now we are coming to these myofilaments, the smallest division of the muscle. You know, if you broke it down from muscle, now we come down to the tiniest thing. So, you know, I was giving an example, like, you remember we had a molecule. Molecules were made of atoms. Atoms then were in turn made of the nucleus and the electrons on the outside. And you had neutrons and protons. And that was the smallest that you could divide it to. So now we've come to the smallest part of a muscle, okay? So these are the myofilaments. So these are contractile proteins. They are proteins which can actually contract. And there are two types of proteins. So you can see here, they are called thin filament or also known as actin filament. This is why looking at the picture is really important, because if you look at a slide really well and instead of just reading, so when you read and you look at a slide, always know alternative names. So thin filaments are also called actin filaments. So you can see it's given right here. And then thick filaments are also known as myosin filaments. So these are the two myofilaments, the actin and myosin. Actin is also called thin. So, I mean, the way you can remember is actin, the thin, that way. Myosin is thick filament. And here now let's see how these bands, we called it A band and we called I band, and let's see what these are made of. And let's also look at some terms that we have here, like, for example, the word sarcomere and H zone and so on. Okay, so just look at this picture, don't write. So let's look at this picture. So here is the banding pattern. So we go back to this previous slide here. So can you see dark band and light band, dark band, light band, and so on? So now we've kind of enlarged it. So here we are looking at, this is a light band, also called I band. Here's a dark band called A band. Then we have light, then dark, it goes like that alternately. So let's see what this I band is made up of. And if you can see here, this actin filament is shown in blue. Can you see that? The thin filament looks thin, right? So can you see this I band here is only made up of thin filaments, because the I band is from this zone to this zone. Can you see that? Only made of thin filaments, right? Am I right? And what is at the center of the I band? Z disc. Okay? Look at this A band. And here if you look, the thick filament is the one that is colored red. So if you look at the A band, can you see it has both red and blue? So the A band has both actin and myosin. Can you see that? A band has both actin and myosin. And what is the center of the A band called? H zone. Very good. Then here, if you look at this, which is called a sarcomere. A sarcomere is what is called the functional unit of the muscle fiber. So the sarcomere is the functional unit, and look where the sarcomere is going. So that's why you can see it's going from this point, from one Z disc to another Z disc. Can you see this? You can ignore this M line. The M line is the centermost part of the H zone, but you can ignore that, okay? So can you see, just from this picture, you can see your I band contains only thin filaments. It has the Z disc at the center. The A band contains both thin and thick. The thin was actin, the thick was myosin. What is at the center? The H zone. The sarcomere, as I told you, is the functional unit, and this is the one that is going to actually shorten. So sarcomeres will shorten. That's how the whole muscle will shorten when the muscle contracts. So sarcomere, you can see, is from one Z disc to the other. So from one Z disc to the next consecutive Z disc. So there are many sarcomeres you can see in one myofibril. And here the same thing is shown. So here if we kind of look at a little bit more structurally at what a thin filament looks like and what a thick filament looks like. So here it's showing you from the beginning of the Z disc. So this is one Z disc, this is the other. So it's just showing you the sarcomere. So you can see here, you can see part of the thin filaments. So they are thin and they kind of twisted around each other. This is the thick filament. You can see it looks rather thick. It tends to be smooth in the center, but towards the periphery, it's got little kind of globules attached to it. Okay? Can you see that? The center is smooth, where this H zone, and in the middle of the H zone, the M line is present. Here you can see one Z disc, here is the other Z disc. And you also happen to have other filaments which are, I mentioned this, you don't need to worry too much about this because it's there in the picture. Someone might want to know what are these elastic filaments. What these are little filaments which are in the core of the myosin. So they go right up to the core of the myosin and they help to restore the shape of the muscle. Remember, muscles can stretch and then it can come back to their original shape. So what helps it to come back to the original shape? It is this elastic filament. Okay? So I hope that's why looking at these pictures becomes really, really important. So I want you to look at these images very carefully. So here, which of these best describes a sarcomere? Very good. It's from one Z disc to the other, not from one A band to the other, because if you look at the picture, from one A band to the other, or from one H zone to the other. So from one A band, so this is one A band to the other A band, you have the I band, not the sarcomere. And from one H zone to the other H zone, you have part of the A band and part of the I band and again part of the A band, right? So that's not a sarcomere. Yes.
Does the H zone separate the two A bands?
No, the H zone is in the center of an A band. See, look at the picture. It's in the center of an A band. It's all one band. It's just a zone in the middle of the band. It's just a zone in the middle of the band, okay? So the Z disc is the sarcomere. So that's why looking at these pictures is really, really important instead of kind of trying to memorize from your notes. Okay, what does the A band contain? Actin and myosin filaments. Yes, not only actin and not only myosin, it contains both. And again, for those who didn't get this right, again, this is where I want you to concentrate on pictures and make sure you look at these slides often. So look at this A band and see here, thick is red, thin is blue. So can you see both thick and thin in this A band? Whereas I has only thin filaments, only the blue color, okay? Where does the Z disc lie? In the center of the I band. Yes, the Z disc lies in the center of the I band. And again, for those who didn't get it right, again, why it's important to look at this. So always have your PowerPoints with you. So here, look at this. It's in the center of the I band, not in the center of the A band and not in the middle of the A band or whatever the other choices were. Okay? Okay, now let's describe the filaments we were talking about, this actin filament or also known as thin filament. It really consists of three kind of proteins in it. So it has two strands of something known as G-actin. And then it has another protein which is called tropomyosin. So again, see the word myosin. So whenever you get these, if a function is mentioned, you must know it. So the function of this tropomyosin, so this tropomyosin is this yellow colored thing. And you can see if you look at this picture up here, this is this G-actin. So just picked out some of this G-actin. These are the subunits. And these are little sites which are present, so which are called the active sites for myosin. What happens is the myosin attaches to the actin. So it'll kind of attach to the actin like this and it'll pull the actin towards the center, and that's how contraction occurs. So these are the active sites for myosin. If you notice, what is tropomyosin doing? It's lying slap bang right across these active sites, okay? So what it does is, in a relaxed state, it actually blocks these active sites. So that's one function. And the second is it also acts as a stiffening agent. It kind of keeps these myofilaments stiff. So this is the function of tropomyosin. So if it's blocking those active sites, how do you unblock them? So you need something to help unblock them. Here we have another protein. So this actin has three proteins, as I mentioned. G-actin was one, which are the blue colored two strands. Tropomyosin is this yellow one. And the other protein is this called troponin, which is this yellow colored one. What it does is, when muscle contraction occurs, it actually binds calcium ions. and it changes the shape. So what it does is, the way when it binds calcium ions, the shape will change in such a way that the myosin will kind of be, this tropomyosin will be pushed away, so that the active sites now will be open and myosin can attach to it. So it exposes the active site, so it changes the shape of this whole actin filament. So it kind of moves this tropomyosin away, and the active sites now are unblocked and myosin can bind to it. Okay, so these are the actions of tropomyosin and troponin, and actin is the actual filament which has the active sites to which myosin will bind.
So tropomyosin is in the relaxed state?
No, they're both present in all states, but the tropomyosin in the relaxed state will actually block the active sites. When the muscle has to contract, you will see a series of steps occurs, and the unblocking is done by the troponin which binds to calcium ions. Yes, Yvonne, you had a question.
I understand, like the purple balls, it moves them around and shape different shape, and it moves that strand. Is that what it's doing?
What happens is that in this state, like you can see here, what is happening is the tropomyosin is going right across those active sites. You can't see myosin in this picture, but imagine you have myosin filaments present here like this, okay? These myosin filaments, they actually attach like this to the active sites, but they cannot attach because you've got this thing across. So when we will see further, a few steps later, we'll see what happens when muscle contraction actually begins. Calcium ions are liberated. They come and they attach to this troponin. When they attach to troponin, imagine as if the troponin kind of changes the shape of this filament. So it kind of moves this yellow thread to another place. So it exposes these active sites. So then now those myosin heads can attach to the active sites, and that's how muscle contraction is brought about. Okay? So here we are just looking at this. You will see how it happens. So we described actin, so now let's look at myosin filament called thick filament. So again, like I said, I can't emphasize enough, you must know these different names. So each thick filament really consists of many myosin molecules. So you can see many of these myosin molecules, they have a tail. So you can see each molecule is really made up of a tail which is smooth, and it's got two of these heads. So now when many of them come and attach to each other, many of these myosin molecules attach to each other, they form a myosin filament. It so happens that all the heads are present towards the periphery and the tails are present towards the center. So with the result, if you looked at, I'll show you this picture. See, can you see this picture here? Can you see all the heads are at the periphery, the tails are in the center, which is why the central area is smooth. Okay? So that's what this is saying, that the tail, it has a tail, and each filament, each molecule has two heads. So many molecules together, you can see many heads, and that's what constitutes a thick filament. In the heads, you have sites for binding actin. Remember, we just talked about actin and we said it has myosin binding sites. So obviously this myosin head will have an actin binding site. And it also, we will see that ATP is needed, because when any work has to be done, you need energy. Remember when we were doing chemistry, we talked of ATP, which is the energy source. So it also has a site where it actually binds ATP, gets energized, and then it is able to bind to actin. So these are the two parts to the myosin head. And as I said, the center of the filament is smooth because that's where the tails are present. Some more terms that we are going to do. So again, here you have to know these two terms, terminal cisternae and what we are going to call T-tubules. You must know what they are part of and what is the function. So here again, we are looking at a muscle fiber. This is a muscle fiber. And remember, it was made up of many myofibrils. So here it's showing you just one part. So here is the sarcolemma. These are the myofibrils. And I told you earlier on that each muscle cell is very similar to any other cell in the body. It had nucleus. It has many nuclei. So here are the nuclei. It has mitochondria, which you can see over here. I told you it has endoplasmic reticulum. It will have all the other things which a cell has, but we are not worried about the Golgi apparatus and the other things that there are there. But we have to look at something called the sarcoplasmic reticulum. Instead of calling it endoplasmic reticulum, here we call it sarcoplasmic reticulum. Remember the word we used was sarcoplasm. So the endoplasmic reticulum, which is here, can you see this is the endoplasmic reticulum which is present here. This in a muscle cell is known as sarcoplasmic reticulum. The word reticulum means network-like. So if you remember when we were doing endoplasmic reticulum, we had a smooth and a rough. They were network of channels, right? Like a network. So can you see this area here is the sarcoplasmic reticulum. This area here is called sarcoplasmic reticulum. So it's like a network. What happens to this sarcoplasmic reticulum? If you just look at this picture here, if you look at this picture and you take the sarcoplasmic reticulum around the center of a sarcomere, this sarcoplasmic reticulum is kind of like a network. When it kind of goes towards the periphery, at the junction of the A and the I band, this sarcoplasmic reticulum kind of gets dilated. So that's a dilated channel which kind of runs perpendicularly like this, it's running in this vertical fashion. So can you see this dilated channel here, and you can see this dilated channel here? Then this would be somewhere here would be the sarcoplasmic reticulum of the next sort of AI junction or next sarcomere. Again, here you have this dilated channel, here again you have dilated channel, here on this side you have on this side. So these dilated channels which kind of run in a perpendicular manner or run vertical to the long axis of the muscle of this myofibril, this dilated channel is known as terminal cisternae. How are you going to remember? The word cisternae means dilation. Cistern means dilated. Cisternae means many. So this means like, let's say, some sort of a dilation, some dilated structure. Terminal towards the end. Can you remember that? Okay. So terminal dilated channel. So you can see here, so this area here, this sarcoplasmic reticulum, the same color as it's going towards the ends of the AI junction, that's where you can see that here it becomes dilated, and that dilated part is known as terminal cisternae. Don't think too much about it. It's nothing but just the dilated portion of sarcoplasmic reticulum. Okay?
Is that like the light gray color?
No, it's this blue. It's still the blue. It's the blue color. The gray is something else.
Okay.
So that's why, look at it. See, terminal cisternae. See how it labels it to the blue. That's why it's very important to look at the picture. The function of this terminal cisternae is to store calcium ions. Remember, I just mentioned calcium ions and how it was important because that troponin bound to calcium, and that's how it helped to unblock those sites. So this function of this terminal cisternae is to store calcium ions and release it when it's needed. Next structure is something known as T-tubules. T stands for transverse tubules. It's called T-tubules because they're also known as transverse tubules. Now imagine so here if you look at this picture, suppose you had something like this, okay? A straight line, a straight cell membrane. And then you took the cell membrane and you kind of made it go in like this and go out like that. Can you see that now you have, in the same area, you have more surface? So this kind of a thing where it goes inside is known as an invagination. You all remember you did microvilli when we did cell. And if I was to draw a microvillus, it was an evagination because this is a microvillus, right? It's from a protrusion of a cell. And an invagination is something going in like this. Okay? So an invagination increases the surface area because the cell membrane has gone in, dipped in and come out. What also happens is that you can see this, if this is a muscle cell, or I don't even need to draw it. If you just look here, if this is a muscle cell, this is the sarcolemma, the outside layer. This is the sarcolemma. Can you see any of these, this area of a myofibril, this area of a myofibril, all these areas of the myofibril which are deep inside, they do not touch the sarcolemma. Can you see? The sarcolemma is the area which gets stimulated. Now, if it is getting stimulated, only this outer parts of the myofibril which are in touch with the sarcolemma would get stimulated. So, in order to make sure that everything gets stimulated, what this sarcolemma does is it actually sends in little channels. So, it has these little transverse invaginations which go in and come out, go in and come out. So, it makes sure that this area kind of touches all the myofibrils. So, this invagination is known as a T-tubule or a transverse tubule. What is it? It's an invagination of the sarcolemma. And where is it present? Notice it is present right in between the terminal cisternae of one side and the other. Can you see that? So, it's present very close to the terminal cisternae. And it is important because, as I just said, when a nerve, we will see that the sarcolemma gets stimulated by a nerve impulse. So, a nerve impulse kind of comes and stimulates the sarcolemma, and then it does its job of making the actin and myosin kind of touch each other or attach to each other. So, now because it's very close to the terminal cisternae, what it does is when the nerve impulse comes, it stimulates the terminal cisternae. What do they have inside them? Calcium ions. So, they release the calcium ions. Those calcium ions then go and get attached to that troponin. The troponin then turns and unblocks, and then the myosin can attach to the actin, and you can see that the filaments can contract. They can slide and they can contract. Can you understand the reason why you need this T-tubule to be present? Okay, because you need something to stimulate calcium to be produced, to be released. So, that stimulus comes from a nerve impulse which travels down the sarcolemma, okay? So, we will see all this in future slides, but for now, I'm kind of giving you a brief thing. So, for now, what you have to remember is, what is the terminal cisternae? So, as I said, remember, cisternae means dilations. They are at the ends, they are parts of the sarcoplasmic reticulum. They store calcium and they release it when it's needed. What is the T-tubule? It is an invagination of the sarcolemma. There's some way you've got to remember which is which. Okay, so if you can come up with a good way to remember, let me know. I'll tell you all. I've racked my brains. I can't come with a good way to tell you how to remember them. They are very close to the terminal cisternae. So when the sort of nerve impulse passes by, they help to stimulate that. And we have another term. You can see that there is this one gray band here, which is the T-tubule. And on either side, you have the terminal cisternae. So you have three structures together, and that is known as a triad. Triad meaning three. So two terminal cisternae and one T-tubule in between. Followed? So when the nerve impulse arrives, it kind of makes a change to the sarcolemma. That change is known as depolarization, and we'll see that later, and that's what kind of continues forwards, and then it stimulates the terminal cisternae to release their calcium ions, and then we'll see something called sliding of the filaments occurs and contraction will occur. So does anyone have any questions so far? It's a lot, I know, it's a bit
Can you say what the exact function of the transverse tubulus was?
The function of the transverse tubule is, because it is an invagination of the sarcolemma, it's lying very close to these terminal cisternae, right? So it's when it gets stimulated, it sort of stimulates these terminal cisternae and signals them to release calcium ions, because that's what they store. So there's no point just storing them. They have to release it. In order to release it, they need something to stimulate them. What stimulates them is the impulse which has arrived at this T-tubule. Okay? So here it'll probably become a little bit easier. So here just listen and watch, because I have a nice sort of video which shows you this. Okay? So let's see, we talked of myofilaments. We talked of these myofilaments. These were thick filaments or myosin. These were the actin filaments. This here was the Z disc on this side, the middle of the I band. Here's another Z disc, the middle of another I band. So this area was the sarcomere, which we said was the functional unit, okay? I also told you that this actin had those active sites on it to which myosin would bind. And when we did the myosin filaments, we said the myosin had the opposite. They had actin binding sites. So what is this binding all? Why do you want to bind? So this is very similar to a zipper-like action. So, as you kind of pull up a zipper, so imagine if myosin filaments attached here like this. They attached here. What will happen? They pull the actin towards the center. Can you see this? So when they pull the actin first towards the center from this position, the Z disc will move here. Right? Then they detach because they use ATP. They detach and then they attach again. So they pull the actin filament even closer now. So now the Z discs come this close. Can you understand? Okay? So as they keep coming closer and closer, finally what happens is that you can see here at the end, when they have come as close as they can come, can you see that the actin now here at this point, the actin was overlapping the myosin only to a certain extent. In this picture, can you see the actin is overlapping the myosin totally, and they actually even overlap each other to a slight extent, right? What else do you notice in this picture? Can you notice that the Z disc from this position has moved to this position? So the sarcomere has shortened. If all the sarcomeres shorten like this, so it's like an accordion, right? If all the sarcomeres shorten, the entire muscle will shorten because finally it's the muscle, right? So the muscle will shorten when it's contracting, okay? So what happens here? The Z discs approach each other. The sarcomeres shorten. The I bands also shorten because they're pulling the Z discs with them. But the A band remains the same length. Can you see this? The A band remains the same length, but the I band shortens. And the H zone, which was the smooth area in between, that disappears because now there is overlap by the actin filaments, right? The H zone was the area where no actin was kind of overlapping. Now, during contraction, the H zone kind of disappears, okay? Have you followed? So this process where these filaments slide against each other is known as the sliding filament theory or the sliding filament mechanism. What does it do? It brings about contraction of the muscle because the sarcomeres shorten, the muscle contracts and the muscle shortens. Okay? So the function of the myofilaments is to form these attachments, and those attachments where the myosin hangs on to the actin and pulls it, that attachment is known as a cross bridge. So the attachment is called a cross bridge. So when they form the cross bridges, contraction begins. Then they begin to pull the actin filaments towards the center. So the Z discs will approach each other. When the Z discs approach each other, notice this was the length of the sarcomere. See what happens. The sarcomere shortens. The I bands also shorten. This was the I band. So notice here the I band is also shortened. The A band remains the same length. And this H zone disappears because all the filaments now completely overlapping each other. Okay? So let's take a look at this.
This is happening all the time.
This is happening when your muscle contracts and it shortens, or not when it's in a relaxed position. So say, for example, there was a book on the table and you wanted to lift that book like that and your biceps muscle was contracting. This is what is happening. If you could actually peep inside and look, really, really microscopically, this is what is happening.
But not when
Not when your muscle is relaxed, then you're in this stage. Right? This is when it is contracted and shortened. Okay?
Oh, I said fuel.
Yes, you had a
So
question?
I just wanted to verify the I band was represented by the yellow.
Let's see, the I band.
Yeah.
1 second, let me just,
I'm pretty sure you went over it already.
let's look at this. The I band is this area, this blue line till this part. Okay? 1 second, let me, oh my God.
My future colleagues look up from the PCR machine. Checking me out, man. PhD's on the scene. So I claim my pipette and pop it up to hold the leaving. The thermocyte that go in my samples called chilling.
Yeah, 1 second, let me just.
My life, the instrumental
Okay. Yes. So here, the I band, if you notice, the I band is showing you this from the end here, where the beginning of this myosin is, from this point to the other end of the actin filament. This yellow thing which you're seeing is those elastin filaments. So ignore the yellow. The I band is, we're only concerned with the actin filaments. Okay? Okay, now I've got to do this again. Okay, so here now let's look at
this scene shows the molecular relationships of actin and myosin relative to the banding pattern of the sarcomere and demonstrates how this relates to sarcomere contraction. Note the relationship of actin and myosin molecules relative to the banding pattern that we have discussed for the sarcomere. Myosin filaments consist of cross-links that bind to specific receptor sites on the actin. The cross-links pull the actin along the myosin.
So can you see how they're kind of being pulled towards each other?
Note that the cross-links attach and release independent. If the cross acted in unison, they would all be released and the myosin attached from it allows the sarcomere to go back to its uncontracted state. Let us look more closely at how the cross-links slide the actin along the myosin.
So there, the actin, all them kind of trying to say it's do not detach at the same time. Well, one is attaching, the other one is detaching. You know, when the other one is attaching, this one is detaching. So that at any one given time, there's something pulling it towards the center. So when they approach the center, when the actin filaments come to the center, you can see the myosin now is completely overlapped, so the H zone disappears. And the sarcomere also shortens. Okay?
Yeah.
It's maybe like a tug of war or... yeah, but tug of war is actually, no, you kind of pull one away from each other. So this is a tug of war going on at both ends, you know, kind of pulling it towards the center. Okay? So let's stop here.